Good morning, everyone. And it is my great pleasure to introduce Dr. Jonathan Kirk from Loyola University, Chicago. And he is currently Associate Professor in Department of Cell and Molecular Physiology, and also directs Loyola Proteomics course. And he received his bachelor degree in bioengineering from Washington State University and a PhD in bioengineering from University of Pittsburgh. As a bioengineer, he studies cardiac mechanics and have published many fascinating and exciting discoveries on why the heart fails as a pump. He has shown that cardiac metabolic change can directly modify myofilament and therefore affect cardiac mechanics. He has servings in many societies and editorial board and also NIH study sections, and is well funded by NIH, DoD, American Heart, and other organizations. Today, he's going to tell us how the sarcomere cycles, and think that's an important component about the cardiac contractile apparatus, how they get repaired. Thank you, Dr. Johnson. Welcome.
Thank you. Thank you very much for the kind introduction and the invitation. Very excited to talk to you all today about some recent work in the laboratory looking at the cardiac sarcomere and how the proteins that make up the sarcomere are maintained during the lifespan of the individual. And the sarcomere is pumping your entire life. It's controlling, it's the engine of the heart, if you will. And so I conceptually have always thought of this as how do you repair the engine of a car, but you can't stop driving the car? So that's going to be the topic of today's talk. Sure, here we go. Just a couple of disclosures before we start. I work with several different drug companies on sarcomere modulators, as well as there is some overlap here with BAG3, but I'm not presenting any work today that is impacted by any of these disclosures. So, like I said, the cardiac sarcomere, made up primarily of actin thin filaments and myosin heads here, represents the engine of the heart. It is almost crystalline lattice structure inside every cardiomyocyte, and it contracts and relaxes with every beat of the heart. And so this constant mechanical strain of contracting and relaxing with every heartbeat will cause those proteins to unfold and denature. And yet the sarcomere, because myocytes are terminally differentiated, needs to maintain this optimal function for decades of life. You can't just tear down the whole myocyte and start again. So you have to be able to replace and repair and switch out these misfolded and unfolded proteins during the contraction cycles of the heart as it continues to beat once a second, more or less, for the entire life of the individual. So this question sort of raised the idea to me of how are sarcomere proteins turned over when you cannot stop contracting ever? So this is sort of the underlying question. All right, so this is the fundamental question that I'm trying to address in my lab, one of the fundamental questions. And thinking through this question conceptually, this actually really reminded me of the 1994 Keanu Reeves, Sandra Bullock masterpiece, Speed. And if you've never seen this really phenomenal action movie, I'm going to explain it to you very quickly here. It's not all that complicated. Basically, bad guy puts a bomb on a bus, and if the bus slows down below 55 miles an hour, the bus blows up. And so Keanu Reeves and Sandra Bullock have to go in and save the day. And so I thought of our question about how are you basically having to maintain the sarcomere, even though it can't ever slow down, as sort of an analogy to this movie Speed. And so personally, I really love movies, and I thought, well, it'd be fun to go through the BAG3 story here as if we're going through a typical action movie. So we're going to start off, of course, any good action movie. In the first act, we'll introduce the stakes. So what happens when you don't turn over the sarcomere and in an appropriate way. So whatever this mechanism is, we haven't defined it yet. If you can't replace these constituent proteins and turn them over with new folded, correctly mechanically active proteins, what happens to the heart? So our story is going to begin back in 1967 when the venerable Eugene Braunwald published this paper in Circulation Research, back when you really only needed a couple figures to publish in Circulation Research, and found here, I've sort of highlighted the important text from the document, was that they basically found that in an animal model, this is a cat model here, of heart failure, there was an extreme depression of the intrinsic contractile state of the cardiac muscle. So in heart failure, and this was shown to be true in human heart failure, of course, as well, the muscle itself is actually weaker. So the heart is failing as a pump, and that's what's a major contributor to the overall disease of what we typically think of as heart failure. Obviously, for other specific etiologies like HFpEF, this may not actually be as true. So, but if we think of the muscle itself becoming weaker, as occurs in many forms of heart failure, we kind of want to wonder, well, where does this behavior derive from? And of course, I said before that the sarcomere is the engine of the heart. And so the sarcomere is what I study, and that's what we're going to be talking about today. But I do want to acknowledge, of course, if anybody in here studies other aspects of the heart, please don't yell at me. I do fully recognize that calcium handling is extremely important. Calcium coupling to the sarcomere proteins here by binding to troponin and activating it. So the calcium handling inside the myocyte is very important, as well as ATP. So the mitochondrial metabolic aspects of the heart are also affected by heart failure, and ATP feeds the myosin ATPases to generate contraction. And there are other elements as well, such as microtubules and protein aggregates that also build up inside the cells in heart failure and can also impact the ability of the muscle to contract. However, as you can see here, all of these different aspects in the cell, all of which known to be affected by heart failure, all feed into the sarcomere as this central regulator of the contractile strength of the myocardium. And so that's what we're going to be focusing here, is the sarcomere itself. And before I go on, I'm going to have to teach you a little bit about this assay that we use to measure the ability of the sarcomere functionally. And you're going to see these data a lot, these type of data a lot throughout the presentation. So I kind of want to go through it nice and slow so that we're all on the same page as far as what we're looking at when we see this data. So one of the experiments that we can do in the lab here is to isolate individual myocytes, shown here as a picture. And we can do this from frozen tissue, because what we do is we expose it to a detergent, Triton X100 here, that skins the myocyte. So it permeabilizes all of the membranes. And so we're really just left with the lattice structure of the sarcomere and the cytoskeleton itself. Then we can take that cell and we glue it to a force transducer pin. So it's attached to this pin on the side, only a bit is shown, and a high-speed length controller. And thus we can measure then the force produced by the sarcomere lattice as we then increase in the bathing solution the amount of calcium that activates the thin filament. Remember, we permeabilize the membrane, so we can control the amount of calcium that is activating the cell. And so we introduce more and more calcium into the bath, which activates the thin filament, allowing myosin to bind actin, creating force-generating cross bridges. And we can measure greater and greater forces as we introduce more and more calcium until eventually we've saturated the amount of force that the cell can produce. And we get data that look like this, which we can then fit to a Hill curve that looks like that. And so you're going to see these curves throughout. Now, from this fitted curve, we can get a number of different parameters of sarcomere function. One of these is the force generating capacity of the sarcomere, or Fmax, so force max. And that is the force that is generated at these saturating calcium conditions. Now, normally, physiologically, you don't reach these saturating calcium conditions inside the myocyte, but it represents sort of a gain factor on the entire curve. And we're going to be talking about this force generating capacity pretty exclusively today. Now, you can also have a leftward, rightward shift of the force calcium curve, shown here as a desensitization to calcium. Now, the mechanisms that control calcium sensitivity are pretty well known to be thin filament phosphorylation of proteins like troponin I or myosin-binding protein C. And we're not going to talk that much about EC50 or calcium sensitivity today, although it will come up one time later on in the presentation. So refocusing back on Fmax here, one of the earliest studies on this Fmax parameter was in a circulation paper from Peter DeToom's lab in 1997. And you can see here in a rat model of heart failure, they saw a pretty robust decrease in this maximum force parameter in the sarcomeres from these animals. And since that original study about a quarter century ago, this has been shown over and over again. So here's a paper from JAK BTS in 2020 showing about a 40% decrease in Fmax from both left and right ventricular myocytes in human heart failure. So here's non-failing, here's heart failure. This has been shown in pig models of heart failure. This has been shown in MI models, in mouse models, both MI and TAC models. And this is a study from me back in 2015, also showing this extreme depression in Fmax in a dog model of heart failure. So everything from rodent models to large animal models to human heart failure all shows this rather extreme depression in Fmax in various models of heart failure. And a lot of times, sort of quantitatively, you can see this is about a 40 or 50% reduction. And so we started off our study wanting to address this question. And so this is now our data, moving from background into the data from my lab. And this is our human cohort from our biorepository here at Loyola. And we were studying 9 non-failing hearts and 21 non-ischemic heart failure or dilated cardiomyopathy hearts. And you can see here there's not really any difference between the two groups, except for, of course, their ejection fraction. And this is, of course, human. If you look up in the right-hand corner, I have a little logo here showing that this is in humans. This will change depending on which species that particular slide is in. So if it's not clear if this is in mouse or in human, you can just look up in this corner and take a look at what species it's in. However, if you're watching over Zoom and my face is up here in the corner, that does not mean that these experiments were all done in me. Okay. So taking a look at this data set here, here in the black curve is the force calcium relationship for non-failing healthy human left ventricular myocytes. And then in the red here is the dilated cardiomyopathy heart failure myocytes. And you can see here when we show the Fmax parameter specifically, that there is about a 40% reduction in Fmax in these human heart failure samples. And up here is a fairly significant p-value. And again, this isn't necessarily surprising. I just showed you that for the past 25 years, we've all shown, various labs have shown this in all sorts of animal models as well as humans. This is probably the best powered study so far to show this, but it's not new. But what isn't known, despite all of these findings for the past quarter century, as well as ours, is why there's a decrease in the force generating capacity of the sarcomere in heart failure. And so that's what we're setting out to address here, is to explain this, this repeated observation in humans. And so one concept, one possibility of this is that the sarcomere itself is structurally disarrayed. And this may be perhaps linked somehow to the protein quality control of that sarcomere. Here is an electron micrograph of an individual sarcomere in a control healthy dog. And you can see the beautiful structure, I think, of the Z discs, the thick and thin filaments, the M line. But if you look at this, at a sarcomere in a heart failure dog, you can see here that you have a loss of the structural integrity of the Z disc. The thick and thin filaments no longer perfectly interdigitate. You have gaps in the lattice and so forth. And it's easy to imagine that this would not be able to generate the same amount of force. It would have less force generating capacity than a healthy control dog. And so we came up with this sort of hypothesis of what might be happening. And if you think in a healthy heart, either human or animal model, you have the sarcomere, and those constituent proteins need to be maintained throughout the life of the animal or human. And so you need to be able to mark proteins for degradation, and that's usually done by ubiquitinating them. And those ubiquitinated proteins can be refolded and chaperoned back into the matrix, or those misfolded proteins, the ubiquitination can mark them for either autophagy or for the ubiquitin proteasome system. So these are systems for clearing out these large proteins, macromolecules that the sarcomere is made up of. And the hypothesis would be then in end-stage heart failure, you have an increase in stress driving more protein unfolding, more protein ubiquitination, marking these for degradation, but that you're not being able to clear those out. And so a decrease in autophagy mechanisms, a decrease in the UPS would cause these proteins to aggregate and stay in the sarcomere structure itself and cause dysfunction. And so this was just our hypothesis of why heart failure might be decreasing the force generating capacity of the sarcomere. And the rest of the study is going to be looking at to see whether or not this is the case. All right. We took our same human heart failure samples that we looked at functionally in one of the previous slides, and we took their left ventricular myocardium and enriched for just the myofilaments, so just the sarcomeres. And because the sarcomere is such a crystalline structure, it stays together. The affinity for the proteins are very high, and therefore it's quite easy to take a myocardium and essentially remove everything else and spin down this very dense, very large sarcomere structure and enrich for the myofilament. And so that's what we did here. So when we just look at the myofilament in the left ventricular myocardium of these human samples, you can see we stained for the protein moiety PTM ubiquitin, which again marks proteins for degradation. And you can see compared to the non-failing controls, the heart failure samples had significantly elevated ubiquitination levels. And you can see that quantified over here, indicating that these proteins are being marked for degradation, but aren't being cleared out. So this sort of matches our hypothesis that we're not, our protein quality control mechanisms in the myofilament are down-regulated in heart failure, and we're having a increase in proteins that should be degraded, but aren't. Now, the question is what's happening to these proteins? A lot of times when we think about depressed protein quality control mechanisms in the heart, we think about big protein aggregates where these proteins are being shuttled away and bind to each other because they're not, they're misfolded and their binding partners aren't well regulated. So we wanted to look at what was happening with these misfolded or unfolded ubiquitinated proteins in the sarcomere. So we did super resolution microscopy for this ubiquitin moiety. And what we found was that these proteins that are marked for degradation are staying embedded in the sarcomere lattice itself. And so these proteins are being misfolded in the engine, in the sarcomere, but they're not being removed. And so they're staying in the protein structure. And we hypothesize that's where they're causing dysfunction. And thus we're getting a dysregulated motor system inside the muscles because we're not being able to remove these proteins that should be degraded and replaced with new ones. And they're just building up there. We wanted to know what these proteins were. So we see that they're in the sarcomere, but are they really sarcomere proteins? You could have a lot of stuff misregulated. Maybe the misfolded proteins are aggregating at the sarcomere just because it's there and it's big. So what we did here was we took non-failing and heart failure human samples again. We enriched for the sarcomere proteins. We digested them with trypsin to generate peptides that we could identify by mass spec. And then we did an enrichment for the ubiquitinated peptides using a diglycine-lysine antibody. The problem is that trypsin removes the actual ubiquitin PTM. And so it leaves behind this diglycine-lysine that you have to identify this way. So you can't identify the ubiquitination modification by traditional mass spec. So once we had identified these, once we had pulled down these ubiquitinated peptides, we analyzed them by mass spectrometry. And what you can see here is just a heat map showing very little ubiquitination in the non-failing samples. And in the heart failure samples, we had a pretty significant upregulation in myofilament ubiquitination. And this was in myosin light chain, desmin, filament C, actinin, tropomyosin, myosin heavy chain, troponin I, actin, myosin binding protein C, troponin C, long story short, we saw that these were the sarcomeric proteins. And so the sarcomeric proteins themselves aren't being turned over, and we've identified these. This protein list of sarcomere proteins that aren't being turned over correctly, we're going to come back to that at the very end of the talk. So just sort of stick a pin in that in your memory, and we're going to come back to that. All right. So we've established the stakes here. Essentially, in human heart failure, the sarcomere proteins aren't being turned over correctly. That the result of that is that Fmax decreases significantly by 40% or more, and thus we have the outcome of the action movie, which the bad outcome here is that, of course, the bus does not survive. Okay. In any good action movie, then you've got your hero. It's introduced in Act 2. And so here we're going to introduce the hero of our story. So see if he can save the day. Beclin 2 associated enthanogene 3, or BAG3. So BAG3 is a pro-autophagy co-chaperone. And by co-chaperone, I mean it binds to a lot of different things. And we're going to go through this here in a second, but the protein structure has all these different binding domains that bring together all sorts of different proteins and chaperones in the cell to essentially coordinate larger signaling pathways. BAG3 is highly expressed in striated muscle, both cardiac and skeletal, as well as the central nervous system, and actually in tumors and cancer cells as well. And in fact, in cancer, it sort of has the opposite impact on outcomes as it does in the heart. We're going to come back to that a little bit later. So here is the BAG3 protein from the N terminus to the C terminus. And if we start here, we have domains that bind to TSC and tuberin. We've got these IPV domains that bind to small heat shock proteins, HSPB8 and HSPB6, as well as HSPB5 or alpha B crystalline, and 14-3-3 proteins here between them. HSPB8 is going to be important for us here in a little bit. We have a PXXP domain here that binds to dynein and therefore is involved in microtubule transport, as well as an SH3 domain containing proteins. And the big one here, which gives BAG3 its name, is the BAG domain. And the BAG domain here binds to Beclin 2, as well as to HSP70. And by its binding to HSP70, it's known to regulate autophagy. All right. Now, like I said, it does a lot of things in the cell. What I want you to remember here is that it binds to HSPB8 and HSP70. And that complex of those three proteins has shown before to be really important for regulating autophagy in the cell. And it was known, it's been known now since about 2006, in this study using a constitutive knockout or heterozygous knockdown, of BAG3, that in diaphragm muscle, if you looked at E18.5 for the mice, the sarcomeres form just fine. So BAG3 is dispensable for the formation, the development of sarcomeres or sarcomerogenesis. But as soon as the animal is born and those sarcomeres are under mechanical stress and strain, i.e. those proteins are being mechanically unfolded and need to be turned over more aggressively, you start to lose sarcomere structure here very rapidly. And so this was just sort of shown early on that this is the phenotype, but mechanistically it wasn't really known what was happening. A couple of years later, it was explored that BAG3 is expressed in soleus muscle here, but in really all types of muscle. And you can see it binding here to the alpha-actinin line at the Z disc. And this paper from Jörg Horfeld's group in Germany showed in a C2-C12 myoblast line that BAG3, if you threw that onto the cytoskeleton, it would release a protein called filamin C. And they identified that here. And so they called this release assay, and used as an example of the need for BAG3 to chaperone proteins out of the sarcomeric lattice in order to be turned over. So this is sort of one of the earliest ideas that BAG3 was involved in essentially chaperoning proteins out of the sarcomere lattice, or the cytoskeleton anyway, to be turned over and degraded to be replaced with new ones. And around the same time, it was started to be noticed that BAG3 was in the heart as well. So this study in circulation research showed that, in fact, it did bind to the Z disc in neonatal rat myocytes as well. So not adult, that wasn't known yet, but in immature cells, it was. And that similarly to that diaphragm study before, that it wasn't necessary for the formation of sarcomeres, but once you started putting those sarcomeres under stress or strain, BAG3 was necessary to maintain that structure. And more recently, it's been appreciated that BAG3 is a very important monogenetic cause of dilated cardiomyopathy. So this study, this landmark study from Dominguez et al. Was published in JACC in 2018, and they identified 129 mutation carriers for BAG3 mutations in a multi-center study in Europe, and followed these 129 BAG3 mutation patients over about 38 months. And you can see here that essentially this is a pretty severe phenotype in patients who have mutations in BAG3, roughly by 15 years, less than 50% survival rate in these patients, and high penetrance of these mutations as well. So even by less than 20 years of age, about 50% of BAG3 mutation carriers have a DCM phenotype, and that by the age of 60, it is over 80% have this heart failure-like phenotype. And some of these mouse models have been created for this BAG3, these BAG3 mutations. Here is a study from Zhu Chen's group a couple years ago, showing in a mouse model of this E455K mutation that it replicates what is seen in humans with really an extreme dilated phenotype, as well as rapid animal death when the animals are still quite young. Okay, now despite the fact that it's involved in genetic heart failure, it is also very critical in non-genetic heart failure. And here's a study from Art Feldman's group in 2014 showing that in human heart failure, there is an extreme depression in the amount of BAG3 expressed in the myocytes from these human hearts. And now coming back to our own work, we've shown the same thing. So this is our dilated cardiomyopathy samples that I've shown you the functional data for, and you can see that there is about a 50% decrease in BAG3 levels in the whole myocardium of these patients. But we're not really interested in the whole myocardium. As I told you, the BAG3 does a lot of stuff in the cell, and we're really focused on the sarcomere. And so from now on, we're going to really be focusing on what exactly is happening with BAG3 at the sarcomere. So here is our data showing in these human myocytes, alpha-actinin indicating the Z discs of these sarcomeres, and BAG3, sort of as we expected, has been shown in skeletal muscle. BAG3 localizes at that Z disc with alpha-actinin very nicely. And when we enriched for just the sarcomeric proteins, we saw the same sort of thing. So here's BAG3 just bound to the sarcomere in healthy human hearts and in heart failure. Now, in the entire myocyte, BAG3 decreased by about 50%. However, at the sarcomere, we only saw about half that level of decrease. So in our heart failure samples, we saw about a 25% decrease in BAG3 levels at the sarcomere. Now, this is just conjecture, but my thought on this, my hypothesis, is that the sarcomeric pool of BAG3 is so important that it's doing whatever it can to maintain as much BAG3 at the sarcomere as possible, which is why even if you have a 50% reduction overall, you only have a 25% reduction at the sarcomere. Now, that's just wild conjecture because I think the sarcomere is so important. I don't necessarily know if the myocyte's making the same call. And if we look now at going back to our functional data in these same humans, if you recall, we saw a decrease in Fmax in our dilated cardiomyopathy, heart failing samples compared to non-failing. But obviously, humans have a lot of variability. So not every heart failure sample saw a significant depression in Fmax. Some of them saw a very mild depression, while others saw a very much larger than 40% reduction in Fmax. So we wanted to know was what role BAG3 might be playing. So we took every human patient that we looked at and we correlated their sarcomeric BAG3 levels, not their whole heart levels, just the sarcomere levels of BAG3 to the Fmax from that individual patient's cells. And what we got was this data here. So in the patients that had the highest levels of maintained sarcomere BAG3 levels, those patients had the highest, almost maintained at normal levels of Fmax. And so if a human heart failure sample had normal levels of BAG3, they also had almost normal levels of Fmax. However, as the patient's BAG3 levels became lower and lower, those patients had the largest or lowest level, the largest decrease in Fmax or the lowest level of function. So there's a strong correlation here between how much sarcomere BAG3 has been reduced in a patient with how weak that patient's heart is. Now, we're going to come back here to calcium sensitivity. I said, so this is that rightward, leftward shift. Calcium sensitivity, like I said, is mainly related to phosphorylation of the thin filament proteins. And it's well known that EC50 decreases in heart failure. So this has also been shown many times. And because its mechanism is known to not be involved with BAG3, we actually saw here that there was no relationship between the sarcomeric levels of BAG3 that a patient had and their levels of calcium sensitivity. So this is just to show that the relationship with Fmax is a unique correlation, not necessarily just related to how sick the individual is or how dysfunctional their sarcomere is generally. A little aside here is in that earlier study I showed you from the European Consortium on BAG3 mutations, they also had done an analysis of male versus female patients. And here is the prognosis in males versus females. So you can see that the prognosis is worse in male BAG3 mutation carriers than compared to female. So there's a sex difference in BAG3 somewhere. And we decided to take a look at what this might be coming from. And when we examined our own human data, we found that in male heart failure patients, there was this significant depression in myofilament BAG3 levels, but this was gone or much reduced in female patients, sort of recapitulating what was seen here in this larger mutation-based study, that it seems like BAG3 drives a more severe phenotype in males than compared to females. And in fact, if we just looked at all of our samples, non-failing and failing alike, females had a much higher level of BAG3 at the sarcomere to begin with, suggesting they might be protected from changes in BAG3 that might be being driven by these mutations. We wanted to know whether or not this change in BAG3 expression level was being driven by estrogen. So we took an aged rat, gave it OVX to make sure that it was completely estrogen-free, and then delivered estrogen 1 week later exogenously, and then looked at the BAG3 levels in the sarcomere and found that there wasn't any strong relationship between estrogen levels and BAG3. So it suggests that BAG3 expression levels are being driven by an estrogen independent mechanism inside in females. However, when we took our female patients versus male patients and basically looked at their function in those that had high levels of BAG3 and those that had low levels of BAG3, you can see that the impact of having low levels of BAG3 remain the same. So here is the data for male patients showing that if you have low BAG3, you have low Fmax. And we see the same relationship in the females, meaning that what BAG3 is doing at the sarcomere is sex independent. It's the same in males versus females. It's just the regulation of the protein and where it's localizing the cell that appears to be different. Okay, so all of this human data that I've shown you so far is all correlation. That's sort of the name of the game in human studies. Thus, we need to move into animal models to be able to gain a stronger cause and effect relationship here. So in Act 3 of our little action movie BAG3 here, we're going to be, the hero gets into trouble. And so we're going to look at this as a genetic knockdown of BAG3. This is a constitutive knockdown of BAG3 in cardiomyocytes. Now, BAG3 homozygous knockdown is very damaging to the animals. They don't live for very long. I'm gonna show you a piece of data of that later on, but for the most part, we're gonna be looking at just hets here. So a 50% reduction in BAG3. This is showing you that in the whole myocardium, you do indeed get about a 50% reduction in BAG3 expression levels in the heterozygous knockdown, as you would expect. Again, we're interested in the sarcomere. So if we look at just myofilament BAG3, we see that the sarcomere level of BAG3 is reduced by about 25%. This is interesting because this is quantitatively almost identical to what we saw in human heart failure, where in the whole myocardium, there was a 50% reduction in BAG3, but in the sarcomere, it was maintained at about 25% reduction. And we see this same impact here in our BAG3 heterozygous knockdown mice. And in these heterozygous knockdown mice, we looked for ubiquitination levels and found, indeed, just like in human heart failure that had similar reduction in BAG3, we saw about a 30% increase in myofilament ubiquitination. So indeed, when you're specifically knocking down BAG3 in a myocyte, so this is, discounting all the other things that are going wrong in human heart failure, we've now made a single change. We can recapitulate the decrease in sarcomere protein quality control and an increase in proteins that aren't being turned over and dysfunctional. Of course, our main readout here is Fmax. So we looked at the forest-calcium relationship in these different animals and found that in our wild-type healthy mice, we had a forest calcium curve here shown in black. And when we do the heterozygous knockdown of BAG3 alone, we see a similar to human 40% reduction in Fmax when we lose half of the BAG3 in each animal. Okay, so now we know that this dysfunction that we're seeing in the sarcomere, the loss of protein quality control mechanisms, is really being driven by BAG3 specifically and not necessarily all the other things that are going wrong in heart failure. But I told you before that BAG3 is a co-chaperone. It works with other things. So the question then becomes, what other things in the cell, what other proteins in this complex are working together in order to help BAG3 do its job at the sarcomere? And thus we enter Act 4 of our movie here, where we're going to assemble the team. So what are BAG3's partners at the sarcomere? Of course, we looked through almost all of BAG3's binding partners, and we found that in addition to BAG3 being at the Z disc, we also found that heat shock protein 70 and heat shock protein B8, the small heat shock protein, are also both at the cardiac Z disc with a similar localization pattern to BAG3. Obviously, you get more background here just because there's a lot of HSP70 and HSP8 in the cell, also in the cytoplasm and other things sort of hanging around. But a significant portion are with BAG3 at the Z disc. So we did IPs, immunoprecipitation assays to pull down the binding partners and found that indeed BAG3 pulled down HSP70 and HSP70 pulled down BAG3. And subsequently, we looked at BAG3 pulling down HSPB8, and HSPB8 was able to pull down BAG3, all at the cardiac Z disc. Thus, this data together suggests that BAG3, HSP70, and HSPB8 are forming a three protein complex, at least, at the cardiac Z disc. And as we would expect, this complex of BAG3 responds to stress. If it is in fact involved in turning over the sarcomeric proteins in response to those proteins needing to be removed from the lattice, we should get an increased expression of those when the cell is stressed. And this was done in a dish in NRVMs, neonatal rat ventricular myocytes. And of course, we found that if we hit them with the proteasome inhibitor MG132 that caused a buildup of ubiquitinated proteins, we saw increased levels of myofilament BAG3, HSP70, and HSPB8, all of course being driven by this increase in sarcomeric ubiquitination. Thus, this complex is a stress-responsive complex at the Z disc, as we would expect it to be if it's doing what we hypothesize it is. And if we look at the relationship between the different proteins in this complex, we'll start here by looking at our mice. we looked at myofilament levels of BAG3, HSP70, and HSPB8 in our heterozygous knockout mice and our young, before they died, homozygous knockout mice. So you can see here the levels of sarcomeric BAG3 reducing more and more as we increase the knockdown. And, oh, sorry, this is not BAG3. But if we look then at the sarcomeric levels of HSP70 and HSPB8, we find that the small heat shock protein, HSPB8, follows the levels of BAG3 almost exactly, where you have a 25% reduction in the heterozygous knockout and a complete absence of it in the homozygous knockout. Whereas HSP70 does not seem to be necessary for BAG3 doesn't seem to be necessary for HSP70's expression. It's unchanged at the sarcomere, regardless of whether or not BAG3 is there or not. So even though they bind each other, BAG3 isn't necessary for HSP70 to be there. We looked at humans in a similar sort of way, where we took all of our human data and said whether or not they had high levels or low levels of sarcomeric BAG3. And we found that patients that had low levels of sarcomeric BAG3 also had low levels of HSPB8, just like we saw with the with mice. And just as we saw with the mouse, the levels of HSP70 at the sarcomere were unchanged if the patient had low levels of BAG3. So again, the HSP70 levels were independent of BAG3's presence or not. So we wanted to know then, what is HSP70 doing in this complex? They're binding there, but HSP70 doesn't need BAG3 to be there. And in order to study this, we used a compound called JG98. If you recall, I told you when I first introduced BAG3 that BAG3 was a target in cancer as well, because tumor cells have to replicate quickly, and therefore they need to have very high protein quality control mechanisms. And therefore, BAG3 is a target for the treatment of cancer in the opposite way that it is conceptually for the treatment of we would expect for the treatment of heart failure. And so Jason Jeswicki's lab developed this compound called JG98 that is an allosteric inhibitor of the interaction between HSP70 and BAG3, which again, we would anticipate would be helpful in cancer cells, although damaging in the heart. This is just showing that when we use this in neonatal rat ventricular myocytes, that at 1 micromolar JG98 reduced the interaction between BAG3 and HSP70 by about 50%. Using this compound to tease apart in the sarcomere what the importance of the HSP70 BAG3 relationship was, we used this cyclohexamide chase experiment to look at protein stability in the sarcomere in these NRVMs, and found that normally the half-life, cyclohexamide inhibits translation of the protein. Sorry, so when you throw that in, you can look at how long the protein lasts because you're not making any more protein. And so here in normal cells, the half-life of BAG3 is about 30 hours. But if you disrupt the interaction between HSP70 and BAG3 with this JG98 compound, we find that the half-life of BAG3 reduces to about three hours. Therefore, HSP70 isn't required for BAG3 to be there or vice versa. However, BAG3 requires the presence of HSP70 for protein stability. If the complex is not all there, then BAG3 is quickly degraded by the cell. Which brings us then, so that explains this team of HSP70, HSPB8, and BAG3. The question then at the end of any good movie is, can the hero save the day? Can overexpression of BAG3 rescue what we've seen in heart failure? And we're going to do this here in a mouse model. I'm using myocardial infarction. So left anterior descending, we tie that off, cause a large infarct. And then over 8 weeks, the mouse develops heart failure. And then we, using AAV9 gene therapy, we introduce either increased levels of BAG3 or as a control, just GFP. And this is the MIC tag that's on the BAG3 showing that in fact we do get at 10 weeks, after the treatment here, we are getting expression of the BAG3. And it had been shown previously that this BAG3 gene therapy helps overall function. So here in green and yellow show the sham animals. In blue is the MI model with treated with just GFP control. And then here at 8 weeks, when the the BAG3 treated animals get their AAV9, you can see a recovery of function. But we're not interested, again, in the whole heart. We're interested in the sarcomere. We wanted to know, what does this AAV9 gene therapy of BAG3 do at the sarcomere? Thus, here is the ubiquitination status of the sarcomere in sham mice and in heart failure mice. And you can see that we get about a 30 or 40% increase in sarcomere protein ubiquitination. This is similar in magnitude to what we saw in the human cells, as well as in the genetic knockdown of BAG3. See very similar impact on sarcomere myofilament ubiquitination. However, when we overexpress BAG3 levels in these mice, we show that we can recover, not necessarily back to sham animals, but definitely significantly reduce the amount of sarcomere ubiquitination that we see in these animals. So BAG3 is helping to clear out these misfolded ubiquitinated proteins from the lattice. And when we look here at function, we see that in our our mouse heart failure model recapitulates similar, the 40% reduction that we observed in human heart failure, and that when we overexpress BAG3 levels in these mice, we get a recovery of function. So this BAG3, exogenous BAG3, is able to come in, pull these misfolded proteins out of the lattice to be replaced by new sarcomeric proteins, and this recovers and rescues function. We wanted to know this exogenous overexpression of BAG3, what was it actually clearing out from the sarcomere? What proteins was it actually turning over? What were its clients? So we took human heart failure myofilament. Again, this myofilament has increased ubiquitination of the sarcomere proteins and a decreased levels of BAG3. And we hit it with exogenous BAG3 to do a client release assay. But instead of in the previous studies where they just looked to see if it was filament C, we took and analyzed these released clients by mass spectrometry to say, what are all the proteins that BAG3 releases from the sarcomeric lattice? I Googled a picture of clients, and this is just what came up. Okay, so we first just did a Western blot for the ubiquitinated proteins, and you can see here when we treat the myofilament and measure the released proteins, we can see that BAG3 releases ubiquitinated proteins. That makes sense. Those ubiquitinated proteins are being marked for degradation. We don't really want BAG3 releasing non-ubiquitinated proteins because those are presumably still functional and fine. We only want it releasing the ubiquitinated proteins, and that's what we saw here. When we analyzed this by mass spec, we identified there were actually 8 clients of BAG3 released in this human heart failure sample. And many of these were our sarcomere proteins, including alpha-actinin, desmin, filament C. This was nice to see as sort of a positive control for our assay that we're able to replicate what Jörg Hofeld's group had shown previously in skeletal muscle, shows that there's similar mechanisms between cardiac and skeletal. Myosin binding protein C, myomectin, and so forth. And now, if you remember, when we did that first ubiquitinin assay in the sarcomere, we identified a lot of different clients that had increased ubiquitination in human heart failure. And there was a large overlap between those that we identified with significant increased ubiquitination and these proteins in human heart failure that BAG3 is able to clear out. So to summarize, sarcomeric levels of this protein, Beclin-2 associated amphetamine, or BAG3, are decreased in human heart failure as well as mouse heart failure. Although this is a more severe phenotype in males than compared to females. And this decreased depressed BAG3 results in lower levels of sarcomeric protein turnover. And these sarcomeric proteins are ubiquitinated and accumulate in the lattice structure itself, so they don't aggregate outside. They stay in the engine, causing dysfunction. And that results in a decrease in the force-generating capacity of the sarcomere. This complex that's made up of BAG3, HSP70, and HSPB8 resides at the Z disc and has at least 8 sarcomeric protein clients. So it's possible that mass spec missed others, or that the dynamics of it were just outside our treatment schedule of how quickly we analyze these after treating them with BAG3. And we found that overexpressing BAG3 protein levels in heart failure in a mouse model restored sarcomere protein turnover and essentially completely rescued function. So I'm going a little bit long, so I'm going to skip these next couple slides. Although if someone has questions, I'm happy to show it. These are some follow-up studies that we're working on now, looking at other aspects of sarcomeric protein quality control that BAG3 is involved in, including chaperoning the misfolded proteins to autophagosomes. So we have some pretty pictures of how this pathway is altered by the presence of BAG3 or not, as well as other mechanisms of protein turnover in the sarcomere that also involve BAG3, but are obviously very different from what we've talked about in this study here. Most important slide, of course, in any talk is the acknowledgement slide, since this work was not done by me in the lab. A big thank you to the members of my lab, the current members shown here. The majority of the work I presented today was done by Dr. Thomas Martin, who defended just a couple months ago and moved on to do his postdoc. He's shown here, as well as our collaborators at Temple University, led by Art Feldman, as well as our funding sources from the NIH and AHA. And I'd like to thank you for your attention, and I welcome any questions from the audience. Thanks.
Thanks so much for the so interesting talk. It's very, very interesting. So you think that the BAG3 mainly affected the ubiquitination of regulated protein or contacted proteins?
So, I mean, we've shown that it has, it has compound, its clients include both myosin and troponin. So it seems to be important for multiple different elements of the sarcomere. So also both the contractile and the regulatory proteins seem to rely on BAG3 for being chaperoned out of the lattice, as well as we think being chaperoned to the autophagosome to being shipped off for degradation. So that was a piece of data I didn't show. As far as what necessarily makes something a BAG3 client versus not, we don't know that yet. Also, we haven't fully identified all of the clients. I mean, we've done this client release assay now a couple more times and have identified a couple additional proteins that may be clients, but we're not sure. And like I said, we're not sure what necessarily gives that selectivity to BAG3 for what it should be chaperoning out of the sarcomere versus what it isn't chaperoning out of the sarcomere.
Okay. Anyone have questions can just unmute and you can ask directly.
Or the chat box is fine too. I can open that up now. Now that I'm not talking, I can pay attention to the chat box.
This is Dr. Vanessa Dayton. I'm in hematopathology, but I'm curious, do you foresee a future when we do serum testing of BAG3 along with troponins in myocardial injury? So
thanks, Vanessa. That's a great question.
Good. Thank you for saying so. Okay.
It's a very interesting concept. The problem, unlike troponins, I said that BAG3 is actually expressed in a lot of different cell types, and so it's in skeletal muscle, it's in the CNS, and therefore its ability to be responsive to indicate like myocardial specific damage might be low, but because it is so sensitive indicator of stress and damage, it may be that. And in fact, my lab has seen, if you're looking at cells under live cell imaging, when you stress them, if we label the BAG3 with like a fluorophore, we can actually see it leaving the cell. And so we think that this is some sort of cell-cell communication for BAG3 to go out and say, there's damage, there's stress, we're going to send out, to try to help our neighbors. And in fact, there's a group in Europe that actually has a lot more data showing an extracellular communication with BAG3 that may end up in the bloodstream and therefore may make a very valuable serum test. We haven't taken that any further, but I know that they're working on that. And so I think my guess is we're going to all see a lot more BAG3 stuff therapeutically over the next decade or so, because it really is becoming a more and more promising target in all sorts of muscle, as well as neurodegenerative, as well as in the cancer fields. it seems to be a central and important regulator of a lot of cellular functions in all of these different diseases.
May I please ask a follow-up question for Dr. Lee? Do you do we have an IHC for BAG3, and or do you foresee us needing to work that up?
We don't have one right now. I don't know. I think if we have a diagnostic or predictive value, we should be able to validate it.
So have we started looking at validating that?
No, I don't. I think you can propose. I think if there's a good clone or some utilities, we definitely can.
Thank you.
Yeah. I think there's a chat box question from Dr. Klein.
Yes, I see that. The mechanism of anthracycline-related cardiotoxicity, and does it affect BAG3 function? That's actually a good question. We haven't looked at that yet. We are doing some cardio-oncology projects looking at this JG98 compound and whether or not we can, if we treat animal models of breast cancer using this JG98, what the effect is on cancer, which is, at this point, it kills it, and what the effect is on the heart. But we haven't yet looked at other types of cardiac-related damage. It's a really interesting question. We do have, let me see here. I thought I had, maybe not. Oh, I guess I don't have it in here anymore. We do have data showing, so not only in dilated cardiomyopathy, but ischemic cardiomyopathy as well, that BAG3 seems to be decreased in the sickest patients, basically sort of across the board and very different in most types of heart failure etiologies. However, in an acute sense of stress, it's oftentimes increased. And so there seems to be two phases to what's happening to BAG3 levels, where in the earlier stress response, it's increased. And only once the heart starts to really go into failure and mechanisms of response start to become maladaptive, do we see this decrease in BAG3 levels. And thus, it might very well be that with the anthracycline-related cardiotoxicity, that stress on the heart, you may actually see an increase in BAG3 levels at first as the cell's trying to compensate for the damage. Okay, you're very welcome.
And also, I have a related question about the protein aggregate, but also in heart failure, you see loss of myofilament. Do you think any other ubiquitin enzymes maybe increase to promote the degradation of sarcomeres?
Yeah, it's a great question. I think what's probably happening is, again, the heart failure is a continuum, right? So there are multiple stages that are occurring as things get basically go from bad to worse. And the loss of sarcomere integrity you see in sort of at the very end stage of disease. And you don't usually see it in the earlier stages. So what I think is happening essentially is the result of this basically taking out far enough that as the protein quality control mechanisms of the sarcomere become more and more dysregulated and more misfolded proteins, you can't maintain this normal crystalline structure when the constituent proteins are all misfolded. And that's going to then cause this loss of integrity, as well as such disruption to the network, you can't insert new proteins correctly into it. There's not a slot for it anymore because of the loss of all this structural integrity. But that's really hard to tease apart all these individual steps. So we don't have evidence of that yet, but we do know that you get dysfunction, you get a decrease in Fmax before you see that that really massive loss of structural integrity at the sarcomere. So that seems to be, a downstream effect of the loss of protein quality control mechanisms rather than the intermediate step of misfolded proteins, structure, and then function. You get the functional effect is more sensitive to the dysregulation than the structure integrity.
Thank you. I think there's another meeting scheduled for the faculty. So if you have anyone have questions, can email me. I can then forward to Dr. Kirk. And thank you all very much for attending. Goodbye.
Thank you.